Thanks. So my talk will be the fate and effects of small plastic debris in the northern Baltic Sea seafloor. And I hope now when I change the slides, they will actually change. Did something happen? Yes, great. Perfect. Everything is good. Okay, yeah, thanks. So as we all know, the marine litter is a serious threat to the coastal and marine environment. And most of the marine litter is plastic, and currently it has been estimated that millions of tons of plastic waste has been estimated to enter the ocean every year. In the environment, the plastics slowly weather and fragment into smaller microplastic particles. And these microplastics are shown to both absorb and release harmful contaminants. And as they can be ingested by even the smallest animals in the beginning of the food chain, the bioaccumulation and biomagnification of microplastics and associated chemicals in the food chain are creating a lot of concern. I think I'm experiencing some difficulties with the slides. Okay, well, it has been proposed that seafloors would act as the ultimate sinks for most marine plastics. And when microplastics sink to the seafloor, they get in contact with the benthic fauna. And here you can see the most common benthic invertebrates in the northern Baltic Sea, the Baltic clam, the Marencelleria polychaete, and then the amphipod Monoporeia affinis. And bioturbation is the process when these animals transport particles and solutes in the sediment-water interface. And they also transport microplastic particles. And this was one of the things that I studied in my PhD thesis. And this whole presentation is about my PhD thesis, which consists of 4 different studies. In the first study, I studied whether the role or what is the role of bioturbation in transporting microplastics upwards and downwards in the sediment. The second part of the PhD thesis was studying the sorption of polycyclic aromatic hydrocarbons and the development of bacterial communities on different types of plastics. And the fourth part of the thesis was studying the impacts of tire rubber particles on the Baltic clam. And next, I will go into more detail of these studies. So in the first study, now I think there is, okay, thank you. Some problems with the slides, I'm sorry. So in the first study, I used microplastics made from fishing line to study whether the plastics that are deposited to the sediment surface are buried by benthic animals. And this experiment lasted for 3 weeks and was conducted in these types of experimental cylinders. And half of these cylinders contained a community of the previously mentioned invertebrates in natural densities, and half of the cylinders were without any animals and acted as controls. After the experiment, the sediment column was lysed and microplastics were extracted and counted from each layer. Then moving on to the second study, which also was about bioturbation. This was similar to the first one, but this time we wanted to know whether animals could transport buried plastics back to the sediment surface. The plastics were made from Lego bricks, and they were placed in 2 layers in the sediment column. The uppermost layer was in 2 centimeters deep and the deeper layer in 5 centimeters deep. And again, half of the cylinders contained animals and half did not. And this time the experiment lasted 10 weeks. Then into the results. Thank you. So the results of these studies show that 8% of the microplastics residing in the sediment surface were buried deeper in the sediment. And the effect of animals on the microplastic burial extended to approximately 5 centimeters deep, which corresponds to the estimations of bioturbation depth in the Baltic Sea. However, from the microplastics that were placed deeper in the sediment, only around 1% were returned to the sediment surface by the animal activities. These studies together broadly suggest that the Baltic Sea sediments could act as microplastic sinks as the net transfer of particles were downwards towards the deeper layers in the sediments. So when the microplastics are buried, they tend to stay in the sediments. In both of these studies, we also studied whether the animals are ingesting the microplastics. In both experiments, the Baltic clam was the only species that ingested microplastics, and probably because the used plastic particles were too large for the two other species. In the first study, where the microplastics were placed on the top of the sediment, on the sediment surface, 25% of the clams ingested microplastics, whereas in the second study, where the plastic particles were placed deeper in the sediments, only 1% of the Baltic clams ingested them. This pattern is probably explained by the fact that these animals feed from the sediment surface, as you can see from the GIF in the side. And that means that they are probably most exposed to the particles when the particles are located on the sediment surface. And the risk for the ingestion decreases when the particles get buried. Then on to the third study. Can you put one slide backwards, please? Thank you. So not only microplastics, but also various anthropogenic contaminants accumulate in the seafloor sediments. And in this third study, we focused on the polycyclic aromatic hydrocarbons that are present in the sediments. And we wanted to examine how they are sorbed to different plastic types. In addition, we wanted to examine the potential degradation of the selected plastic types by studying the bacterial community composition on these plastics. And in this study, we compared conventional and biodegradable plastics as the use of biodegradable plastic materials are constantly increasing in the society, but we don't really have information on their true biodegradability. And now these results, I first tell you what we did. So we had two conventional and biodegradable plastic types, and they were cut into different shaped pieces, but they had the same surface area. These particles were incubated in sediment and water that were collected from two sites located in coastal Helsinki, and the incubations lasted 14 weeks. Half of the plastics were used for studying the PAH concentration, and half were used for bacterial community analysis. And what we found was that the bacterial community composition on the biodegradable cellulose acetate was distinct from the other plastic types. The bacterial community on this material also contained bacteria that are capable of degrading complex carbon substrates, including cellulose. It's possible that the enrichment of these bacteria is because of their capability to degrade cellulose, but we did not actually see any visible signs of degradation. And when we look at the PAH sorption to the different plastic types, we found that all plastic types sorbed PAHs from the sediments, but in all cases, the conventional polystyrene was the material that had the highest concentrations. The differences in these sorption patterns indicate that some plastics may be more efficient vectors for harmful contaminants. But when we examined the concentrations of PAHs sorbed to plastics, they were clearly lower than the PAH concentrations measured in the sediments. And from here, we could see that the importance of microplastics as PAH vectors to sediment-dwelling invertebrates seem to be low, as the animals are more exposed to the PAHs in the sediment than they are for the PAHs in plastics. Then the final and the fourth study. And here we wanted to study whether the exposure to tire rubber fragments have any negative effects on the Baltic clam, because we know that some ingredients of the tire rubber can be really harmful to animals. So the background of this study is that there was a report published last year in Finland that identified car tires and artificial turfs containing rubber granulates as one of the largest microplastic emission sources in the whole country. And the tire material indeed consists of a mixture of different kind of synthetic and natural rubber polymers, as well as numerous additives. And many of these additives can be harmful, such as metals and PAHs and phenolic compounds. And that's why we wanted to study if they have any effects on the Baltic clam. And we focused on studying biomarkers, and we selected biomarkers that are the key enzymes in the antioxidant defense system of these animals. And the antioxidant defense system consists of many enzymes that together try to compensate the harmful effects of contaminants and the contaminant exposure. And eventually this can lead to the protection of the cells, or if the protection is somehow incomplete, this might cause oxidative stress and oxidative damage as well. And in addition, we also studied the changes in the cell ultrastructure, for example, changes in the cell organelles. So next, I will show how the study was made. So there were 3 types of aquaria. In the next slide, please. Thank you. So we had 3 types of aquaria, and one of them acted as control. One had rubber powder inside a mesh bag, and one had rubber powder freely in the aquarium. And the idea of this setup was to compare the potential effects in the clams in cases where they could ingest the particles, and in the cases where they could not have any contact to the particles, and they would also only be exposed to their leachates. And the study was divided into 2 experiments. The acute experiment lasted for 5 days, and the chronic exposure lasted 29 days. And during both of these experiments, the metals and PAHs from the water were measured, and at the end of the experiment, the clams were taken out and dissected. And now I will show the, can you put the previous slide, please? The results. So as you can see from the GIF, the clams ingested these tire rubber particles. So there you can see a siphon of one clam, and all the black small particles are the tire rubber particles. And multiple metals and PAHs were also quantified from the experimental water, especially in the particle treatments. When we looked at the biochemical biomarkers, some of them did not show any differences between treatments, but in some, the clams exposed to the particles showed elevated stress responses, as can be seen here in this figure of the superoxide dismutase enzyme, where the chronic particle treatment clams had the lowest SOD activities. Then we also found that the chronic leachate and particle exposure induced changes in the cell nuclei and other organelles, indicating that the antioxidant defense system has been overloaded and oxidative damage to the cell structures had occurred. For example, in these photos, these 3 photos, we found that there are disruptions in the mitochondria, which are marked with stars, and some disruptions also in lysosomes that are marked with the letter L, and also found some problems in the gills of the rubber exposed clams. And finally, to summarize in the next slide, The bioturbation was shown to play an important role in the vertical distribution of microplastics in the sediments. And the common benthic fauna in the northern Baltic Sea seemed to facilitate the burial of microplastics, while at the same time reducing the bioavailability of microplastics and promoting their preservation in the seafloor. And since the redistribution of buried microplastics back to the sediment surface was quite small, the results also suggest that the Baltic Sea sediments act as microplastic sinks. We also showed that when the plastics end up to the sediments, the polymer type can influence their fate and risks that they pose to the benthic habitat. For example, the bacterial communities that were found on the biodegradable cellulose acetate was different to the other plastic types and had potentially biodegrading bacteria. But we need further research to verify whether the biodegradation of these materials could really be possible. And also the varying PAH sorption capacities on different plastics indicate that if ingested, their roles as PAH vectors can also depend on the polymer type. But however, compared to the sediment concentrations, we also showed that probably the PAH sorption to plastics is not going to add the PAH burden of deposit feeding animals. And finally, sorry.
Thank you.
Just final remarks that the tire rubber particles were also ingested and caused some elevated stress responses. And if you are interested to hear more, I have included a code to download the PhD thesis from the last slide. Thank you.
Thank you.